All right, well, we've looked at some of the basic types of mutations, and now I want to look at more complex forms, okay? The mutations we've looked at so far are basically point mutations. So when we talk about, something like deamination, or when we talk about going in and have oxidative damage, we're talking usually about some form of simply A no longer bonding to T, but somehow bonds to G, and then you ultimately get a single place, a single point, a nucleotide that gets replaced. We've already looked at other mutations, right? We looked at chromosomal mutations. We looked at deletions and duplications or rearrangement. These are mutations. They're just large scale mutations. The ones I want to look at now, though, are what are known as transposable elements and transposable mutations. Now, they can relate directly back to chromosomal mutations because we're going to talk now about big chunks of DNA getting moved around, okay? And moving areas. And we'll talk about these by elements that can transpose or move themselves around. So what in the world are these things? Well, they are mobile sequences, okay? And they are elements of our DNA that pick themselves up and move themselves, okay? They do it in a couple of ways that we're going to talk about, by copying or by using RNA intermediates, but they can move, okay? This is Barbara McClintock, and Dr. McClintock was at Cold Spring Harbor, and she was working on looking at corn. That first picture that we saw, there were corn, right? And she saw that there were these some of the traits. She was just mapping them like you did fly, location studies, right? Where you sit there and you said, okay, where's this gene found? Well, she's just doing location studies back in the 1940s. And somewhere in all that, she noticed that this locus that was supposed to be on chromosome 6 suddenly was on chromosome 5, right? Or something. And she just said, and she said it moved. Everybody said, no, there's chromosomes. We know what chromosomes are now. That's just nuts, right? So really, her first work was met with skepticism. People said, and to be honest about it, she was a female in science, and in the 1940s, and even maybe today, I'm not sure, but seems like it still persists in some ways. But she was fighting against this established ideas of what was going on. And in fact, what was interesting about it is that Goldsmith had proposed this idea of what were known as hopeful monsters. This idea that as evolution proceeded, there was these new things that showed up, and they just became potentially functionally were these weird new variants. It's kind of like these sports, I guess, that Darwin had talked about. Well, it's 1940s, 1950s. Neo-Darwinianism has come along. Remember, I said that Darwin came along. He had lots of issues with his theories. There was all this infighting about it, and then in the 1920s and 30s, and we'll talk more about why. But after that period, it kind of decreased because of social Darwinism and other things, and Darwin's ideas didn't seem to work. But by the 1940s, we had come back to what's called Neo-Darwinian theory. And most of you, as I've mentioned before, trained as neo-Darwinian theorists. And it was based on the idea of these small changes. Well, all these neo-Darwinians out there going, no, you can't have DNA. We figured it out now. There's non-chromosomes. DNA, looks like this. And you can't have it jumping around on us. It's not particles, right? Well, and by the 1960s, some years later, and she's still working at Cold Spring Harbor, the lac operon system, which you probably learned about, is this regulatory system. Turns out that can move around. And so it was, her work was finally accepted. And it always takes a while to get a Nobel Prize, and it took till 1983, and she was awarded a Nobel Prize for discovering these mobile genetic elements. It turned out, yes, in fact, there are these pieces of DNA that can move. Okay, and sometimes when there's two pieces, they can pick up a gene and move them with them, right? If you're on each side of a gene, they'll move a gene around, and those are known as transposable elements. And she worked in the McClintock building. My late wife actually was postdocing in the 1980s in the Barbara McClintock building, Cold Spring Harbor. It's on the north shore of Long Island, a beautiful little village there, whaling village. And this is a beautiful research site. It's one of the first places that trout were actually hybridized and grown commercially in the U.S. Was here, and she was working there in the McClintock building. So I was fortunate enough to meet Dr. McClintock, as well as James Watson, Watson and Crick was the director at the time, and so actually I had a couple chances to meet him as well. But anyway, she was working in Cold Spring Harbor throughout her career for most of the time. Okay, so what is a transposon or a transposable element? Well, it's a piece of DNA that can move around, okay? They're called mobile DNA elements or transposable elements. They've been linked to some interesting theory. One is called selfish gene theory or selfish DNA theory in this case. Remember I told you about selfish gene theory, this idea that genes only cooperate with other genes to kind of get themselves advanced and around and moved on, kind of like probably Congress or something, I don't know, or Parliament. You only cooperate enough to get what you need out of it. Well, selfish DNA is even more than that. They only copy themselves. They're only there for themselves. Literally, they don't have any major role. They're not, cooperating with other genes. They're simply just using our DNA or the animal's DNA or the plant DNA just as a house, someplace to stay. And when they move, it's called transposition. Okay, and this is when they either cut themselves out of one site and move to another, or they copy themselves and make a copy in some other site. Okay. They probably played a role because they can pick up big pieces of DNA and move them. This whole idea of exon shuffling. Remember when we talked about what a gene looked like? We said eukaryotic genes have introns and exons, right? And I said, well, one of the theories, at least, about why we have all these exons or have all these introns is that once you evolve a left turn or right turn or all that, why re-evolve it? Why not just pick it up and make a copy? Well, one of the ways you can pick it up and make a copy is to have a transposable elements inside of the introns, and those can literally pick it up and move it someplace. And we know that there are quite a few there are transposable elements everywhere, and they're certainly found inside of introns. So exon shuffling theory fits in that. Well, these transposons fall into two big categories which have multiple subdivisions, but for us, we're gonna worry about the two. Okay, they're called class 1 and class 2 in many cases, but for us, we just have what are known as DNA transposons or transpositions. Okay, and we're going to have what is known as retrotransposons. Okay, the big difference is that the DNA transposon simply cuts itself out or gets removed, and it physically as a piece goes somewhere else. So the former site kind of goes back together. What we're going to see, this is known as cut and paste, right? You know how you do that, right? You're typing in Word or wherever you've got your word processor, and you cut over it, copy that whole thing out, and I should have used that sentence down here, and you paste it back in. Okay. Retrotransposons actually stay in place, but they produce an RNA through RNA polymerase. They make an RNA message essentially. And then there's a special enzyme known as reverse transcriptase, which is part of a cell anyway. Reverse transcriptase, what it can do is read the RNA and then actually make DNA from it. That's why it's called reverse transcription. And it then makes this DNA intermediate that can insert. Okay, this is known as copy and paste form. Okay, most in bacteria and other things, most of them are DNA transposition, so they don't use an intermediate. But eukaryotes, particularly mammals, use a lot of retrotransposition. So we have lots and lots. In fact, it makes up a huge amount of our genome, which we'll talk about in a moment. Now, in general, transposable elements have sort of a common part. In the middle is sort of genes, their own actual genes, okay, some coding regions. And then they have these, what are known as terminal and flanking repeats. The terminal repeats are areas that tend to get cut out with them and are part of what gets moved. The flanking repeats are areas where they insert in various ways, okay? And these are known as inverted repeats. You can see why they're called inverted, because they're backwards to this other. They're exactly the same. This TGCG is exactly the same as over here. It's just inverted going back, right, on this side. And then the flanking repeats have special recognition, and this is just one example of that, okay? All right, well, the class 1 these are the retrotransposons. There are a number of different forms of these, but for our purposes, we're going to talk about those that are called LTRs. Okay, and LTRs are these long terminal repeats. So they have this, quite a long section here on each end that has this terminal area, and then they have a coding region in the middle of them. Okay, and then we're going to talk about non-LTRs. Okay, now what's interesting about these LTRs, retrotransposons, is they have all the genes they need in there to be a retrovirus. They have everything they need, except they can't make a viral coat. A coat is what's needed to get them out of a cell and to bud, it's called, into another cell. So they can't easily move from one to another. So even though they may have come into us as viruses essentially and started to put themselves into our system, they've lost the capability to actually produce all the genes they need to make a functional virus that might move out. Okay. Now they are pretty interesting, right? They have what's going to happen is you're going to have a start site. You're going to have a copy of that, so you're going to have transcription going on, and there's even a polyase site. These guys can persist, right? So you have a 5' in, a 3' in. They look like messages, and in fact, there are special RNA processing that will go along, and they will convert this into what's known as retroviral genome. They'll essentially make it into a functional piece that can work, and that's part of what goes on up in here. All right, and we'll look at that in just a moment because we need to add in a big piece, which is the piece that can copy it back from RNA to DNA. Okay, now the other parts of these are the non long term, right? And these come up in ALU sequences and all sorts of things, but they're commonly associated with lines and signs. Lines are long interdispersed repeats. These are 6,000 bases long, base pairs long, right? So these are pretty big. They make up a huge amount of our genome in humans. Okay, somewhere over 20% of our genome is made up with these. And then there's short ones, signs, and signs are copied. So this is all that repeat stuff. Remember, that's all over the genome that we have copies of. We wondered way back when we started looking at diversity of the genome and said, whoa, what is all this stuff where it's just everywhere, or lots of it, but it doesn't seem to be genes or whatever. Well, it's a lot of it is just stuff that got in there and copied itself all over the place. And this is certainly one of them. Now this one has only 2 primary domains. It has a first one up here, which has to do with it codes a protein that helps it bind to RNA. That's the ORF. It means open reading frame. It's a gene. Okay, essentially, it's an area that's going to be transcribed and eventually translated. And then it has an open reading frame 2, which encodes for multiple genes. And these are reverse transcriptase is one of them. And there's a DNA endonuclease activity protein that's produced out of here as well. Okay, so they have all of that going on. Now, what happens is the top one is just regular transposon, so we're not going to worry about that. Let's look at a retrotransposon, which is the bottom part here. Okay, retrotransposon, what happens is that it opens it up. It opens up the DNA. It's sitting there. It looks like a piece of DNA to everything else, right? It gets transcribed. It gets transcribed into RNA, which gets poly-A tail and all that. It can actually produce its own protein, okay, because reverse transcriptase. Reverse transcriptase, which is present anyway in cells, reads this piece of RNA and reverses it back from the RNA to DNA. Okay, and then that DNA gets pushed back into double-stranded by replication process. It gets made so it actually reverse transcriptase goes along, makes one strand of new DNA. So you've got this complex hybrid thing here. You got this RNA on one side and DNA on another, double-stranded. And then it turns around and basically it's a complex. Obviously, it's not a single enzyme. Turns around and copies that other side, removes the RNA, and you get double-stranded DNA. And then there is some assistance to get it inserted into a new spot. Okay, so it goes out, makes a measure, goes out and copies itself over here. So you got the original retrotransposon here, and you've got this new copy over here somewhere stuck into place. Okay, so it copies in that form. This is the really, really interesting way that it occurs. Okay, now the class 2s are the ones that again are less common in mammalian systems, but they're present, but they're very common in other systems. And these are ones which they produce a protein known as transposase. Okay, and they sort of help it move through the system in the way. And the way it works is like this, okay? In fact, all of them produce a transposase. Transposase is an enzyme that helps them actually do all this, right? So here we've got it all set up. We've got our transposon here. We cut out the piece that we're going to copy. That piece is then comes along, and what we have to do is we have to make a staggered cut. So and there are all sorts of enzymatic functions that do this. In fact, you can just buy all sorts of things that will recognize very specific sequences, usually palindromic. So they're read the same forward or backwards. This one isn't palindromic, but they'll make a staggered cut. Okay, so here we're going to cut between the T and the A, and the C and the A right here, and then we're going to break all these little hydrogen bonds in between. Transposable element then can come into place and insert itself. Then you've got these little staggered pieces right here, and DNA polymerase comes along and says, "Whoa, I got some openings here. I'm repairing anyway. I must be a mistake." So it looks at what's on the other side, puts a T there and a C there, and fills it all in. Okay. And it basically makes this new form. And so you end up now with this insert actually being in place, right? And so that's basically how it works. Now, with reverse transcriptase, it's the same basic way, except that you make this copy from the message, right? And you read the enzymes that you need from the message to do a lot of this. But once you get a double-stranded DNA, you basically do the same process. You cut it, you insert it, you fill in the gaps, and you've now got it in a new place. All right. You can read and watch this online. There's some really nice details, much more than what I'm doing here. I'm just trying to introduce the idea that these can cause, mutations in some sense in the form we're talking about. So where do we see it? One of my favorite things is wine. I love to travel wherever I go in the world and try their wines. And there are sort of three classic, forms of grapes. They're this sort of what would you call a Concord grape, the wild grape, or forms, and these have lots of anthocyanin pigments, so they're sort of dark colored grapes that you make grape jelly and stuff out of, right? Well, it turns out that white grapes that are used commercially were produced by this happenstance of a retrotransposon, and it didn't insert in the middle of the gene, it inserted close by. Well, close by are regulatory regions, right? But what it basically did was shut down most of the production of this anthocyanin production, this color production, and so you can end up with white grapes like this, okay? And of course, take those white grapes and you make white wine out of it. And red wine, which is what I prefer, is actually made the fact that there was a system that basically cleaved off most of this retrotransposon. Still, some of it's there, particularly some of the terminal repeats, but so much of it got cut off that you now have a partially functional gene that can produce enough of the pigment that you end up cleaning up with what are red grapes, and so here's one of those steps where you had a natural functioning gene, a transposon came in nearby, it didn't move it or anything, it just came in nearby, disrupted the regulatory control. You get one product out of that. When that corrected itself partially, you get a secondary, another product out of it. Of course, all wine's white, and because of internal side of the grapes there, it's just that red wine is the last of the process is to put in the outside layers, and so that absorbs up the red and makes it colorful. Okay. Anyway, that's a nice little example of where retrotransposons have worked. Another place, if you go around, petunias, you see little petunias, very often they've got these little red stripes. Red stripes are the natural color, right? And that's the functional part, and that's where the transposon is absent. But you'll get these little places where the transposon has jumped into the gene, and that's actually turned it off, right? So we end up with this, absence of color type of thing. It's one of those pathways again. Now the transposon has jumped in actually into the part of the gene, and it's no longer producing the product that it can do. So once you don't have that product, you end up white, and you can't put pigment in as you go. So you end up with this classic form. And just a couple other things to tell you, just so you kind of know, one is that you do get some horizontal transfer. What that means is retrotransposons can get moved. How do they get moved? Well, they sometimes get involved in when other viral attacks come or some other pathogen will actually pick up one of these transposons and move it. And sometimes it moves between species. Okay, and that's where we think we've actually picked up some of these. The pathogen will move it to a new species, maybe back and forth between several by a pathogen. So some of these viral attacks actually may pick up when they get into a regular cell, and then they start copying themselves. Well, they may grab a transposon and carry it. So there's some evidence that there is we have exchanged some of these forms. And then it turns out that there's sort of hotspots. Okay, um, I said a huge part of our DNA. So we go back to that highly repetitive, moderate repetitive, all that highly repetitive piece of DNA is a lot of chunk of junk out there, right? It's not all junk. Turns out a lot of it's retrotransposons. Various estimates, I somewhere see 50%. I don't know if that's still correct. Day to day, it seems to go up and down, but a huge amount of our genome is made up of retrotransposons. Okay, and clearly it's been responsible for moving stuff around. Okay, and we know we see these signs and lines and arrows. There was basically clearly an invasion and a duplication going on back in the great apes and our lineage. And particularly when you look at the orangutans, chimps, and humans known as the great apes, um, there clearly was an invasion of retrotransposon somewhere in that lineage, and then once they got there, they just copy to make up a huge amount of our genome. So they played a huge role going back, 15, 16 million years. They've been there in our genome copying themselves, and we know that because we can look at some of the monkeys and apes that have pulled off earlier, rhesus and marmosets and others, and they don't have as many, they certainly don't have the same ones of retrotransposons. Okay, now that we've got them, all kinds of weird things happen. Neurons, there's as many as in each neuron. There's been estimates of somewhere around 80 or so retro changes per neuron. As neurons are developing and copying out, retrotransposons are jumping all around in them. And what's known as a connectome is the idea of how our brains and our neural systems connect. And there's some thought that actually each of our these neurites has a very different amount of duplication of some of these retrotransposons, and they in fact may be controlling how particular neurons work. So it's a really interesting idea. Now there's some counter thoughts to it that no, it's not quite as much as we thought, but there's two schools of thought. But some of the clear data is that we do have a lot of retrotranspositions going on in neural cells, and those neural cells may, in fact, some of their morphology may be driven by the fact that some have more repeats than others, which comes back to why different ones of us can think in different ways, and why I am terrible at much as I love languages, I'm terrible at it. I'm terrible at music. I love it, but I'm just not very good at it. But I'm very good at other things, And why am I like that? And somebody else can pick up a guitar and play it, or listen to music and say that's a C or a G chord or whatever, and I struggle with it. But I can do other things just on top of my head. So why are we wired different? Well, part of the wiring process is that we are wired different, and maybe probably we're wired different is these retrotransposons. We certainly know that it occurs. In things like birds, right? Songbirds. Chickens and things don't have a lot of retrotransposons in their brains or they have them, but they don't see a lot of copies. Songbirds do, though. It's really interesting. The hippocampus of songbirds is really important because that's the area of memory. So a young songbird will listen and hear the sound of a congener of another of its species, or maybe its parent calling, and that's how they pick up and learn to be, the call of a mockingbird versus the call of some other, um, songbird. And part of that process is this hippocampus memory system, right? And in fact, in the spring when birds are really calling, the males are really active, they actually have their hippocampus, their brain grows and changes because of the influence of this. And part of those changes has to do with retrotransposons. And there's lots of other things that go on too, but there clearly are a lot of retrotransposons found there and are active during this songbird time. And then so you know that you always hear you got all the brain cells you're going to have. It turns out that's not quite true. It turns out the hippocampus is potentially an area where it's actually sending out new progenitor cells that can go and become other pieces out here. And we know that because we see these the remnants of that moving through. So it actually is one of the ways that we can replace some neural cells is from our natural process of our hippocampus. And interestingly, the cells that are really close by the hippocampus tend to be at high in these retrotransposons, and others as we get further away, but have generated from the hippocampus actually are less. And they end up with what's called mosaicism. Remember that? We talked about mosaicism early on. We talked about during development, you might have somebody who's, some cells are got chromosome 18 modification in them, but most of the body doesn't, right? That's a mosaic. Well, this is your neural system as mosaics. And so it's another example of why we all think differently. We all have different responses in different ways because we're all truly wired very differently. Okay, well, that's really pretty much all I wanted to say about transposons and those elements, just to make you aware that they are critically important, and they certainly have been over both evolutionary time and potentially just day-to-day activities that go on in humans and in other species. So something to look at. All right, we've got one more lecture to talk about, and it's much shorter than this one. We'll move into that as our last of our mutation lectures.